Hey students, I just wanted to go over signature assignment 3, which was our activity for enzymes in class, just to make sure that we're all on the same track. As you can see here, we start with some definitions and key points, and then we have our first problem that says alpha-glucosidase is an enzyme that breaks down maltose into 2 molecules of glucose. It also breaks down glycogen into glucose. So here we have a diagram of alpha-glucosidase binding to maltose, or maltose I should say, binding alpha-glucosidase on the active site and then releasing 2 molecules of glucose. Maltose is, almost everyone got that correct, it is a carbohydrate. More specifically, it is a disaccharide. What kind of biomolecule is alpha-glucosidase? It is a protein, because enzymes are proteins. This one was a little confusing, but we did go over it in class, so all we had to do is write down the reactant, maltose. Alpha-glucosidase is our enzyme that goes on top of the arrow. And then we have as our products glucose and a glucose. This demonstrates to us that this is a type of catabolic hydrolysis reaction. For E, it tells us the amino acid sequence below is part of the enzyme that is not near the active site of alpha-glucosidase. So not near this part right here. Not here, somewhere maybe over here. Here's our amino acid sequence, and you should have filled in the table from the guide below in a few pages, and what you should have found was that glutamic acid is negatively charged, threonine is polar, proline is non-polar, NP is perfectly acceptable there, histidine is positively charged, and then valine is non-polar. So then it asks you if the his were changed to an arg, if histidine was changed to an arginine, this one right here, do you think that would change the ability of the enzyme to catalyze this reaction? Explain your reasoning. Y'all did some amazing guesstimation in class, and you guesstimated that these are both positively charged amino acids. They're also not near the active site. So most of y'all said yes, it would still be able to catalyze the reaction because his and arg are similar or have similar side chains. Something along those lines. G tells us the amino acid sequence below is part of the active site of alpha-glucosidase. Fill in the table using the amino acid chart. And what we find is that methionine is non-polar. This is polar, negatively charged, non-polar, and polar. And that's just what I'm getting from the table below. H says if the glutamic acid were changed to lysine, do you think this would affect the ability of the enzyme to catalyze the reaction? And most of you said yes, it would affect the ability because we're changing from a negatively charged amino acid to a positively charged amino acid. And we're also changing the structure of the active site. So most of you said that, would this affect the ability? You said yes, it would. Or some of you just answered a different question and said no, the enzyme cannot catalyze the reaction. Or yes, it would have an effect. All right, and that's because we have 2 very differently shaped charged amino acids in the active site. I says if a change in the amino acid sequence rendered the enzyme totally unable to catalyze this reaction, do you think the phenotype of the organism would change? And we figured out that yes, it would change, because now you have an inactive enzyme that catalyzes a reaction to go from a disaccharide. Sorry, I think I have an extra S in there. A disaccharide to monosaccharides. Mono. And that actually turns out to be a fatal disease. It's called Pompe's. Oh my gosh, I can't write. And people typically don't live past a year old. So we're changing the enzyme that breaks down glycogen to glucose, maltose to monosaccharides. And when we get to the end of the semester, we will learn more about the role of glucose in cellular respiration. So the phenotype is the physical expression of the gene. How does the enzyme turn out? Because remember, DNA to RNA to protein. So if we're changing the amino acid sequence by changing, there's some kind of mutation in the DNA that causes a protein to not have the right structure. It's not going to be able to do its function. And remember that structure and function are very related to each other. So this is the flow of information. We're talking about a functional protein being changed to a non-functional protein. I didn't expect you to know about this disease, but I did expect you to be able to figure out that, yeah, if you changed from a negatively charged amino acid to a positively charged amino acid close to the active site or at the active site, this would indeed have an effect. And if you change the amino acid sequence that makes the enzyme totally unable to catalyze the reaction, you would be in big, big trouble. The next part of the assignment is about tyrosinase, and some of y'all downloaded the worksheet from the signature assignment 3, which starts here, and that's perfectly fine. Tyrosinase is an enzyme that helps to convert tyrosine into melanin, the pigment in our skin, hair, and eyes. This is one of the steps in that pathway. In the chemical reaction below, circle the reactant. Bam. There's your reactant. Draw a star or asterisk next to the enzyme and put a box around the product. So what we want you to be able to do is to be able to look at a reaction, label the reactants, the enzyme, and the product. Some people were confused with this one. Is tyrosinase being chemically changed during the reaction? No, tyrosinase is not being changed during the reaction because tyrosinase is an enzyme. If we scroll back up here, here's our enzyme, alpha-glucosidase. What you're seeing is that alpha-glucosidase binds the substrate, releases the products without being chemically changed. So that's the definition of an enzyme. It catalyzes a chemical reaction by lowering the activation energy, and it is not chemically changed during the reaction. B, tyrosinase contains 2 copper atoms at the active site. Copper is a cofactor. Okay, the only distinction we're really making, I know there was some confusion with cofactors and coenzymes. The distinction that we're making between cofactors and coenzymes is that the cofactors are inorganic. Things like magnesium, copper, okay, iron. These are all cofactors, whereas coenzymes are our organic molecules, like the vitamins A, B, C, D, E, K. Those are coenzymes. Now, C says part of the amino acid sequence of tyrosinase is below. So now we're looking at a different sequence, and we're going to fill in the table using the same amino acid chart. Some people were scared because all of them are nonpolar. nonpolar, 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 nonpolar. People were like, is this real? Yes, it's real. So part of the amino acid sequence of tyrosinase is a string of nonpolar amino acids. And then we're going to predict, is this region of the enzyme hydrophilic or hydrophobic? Most of us got this right. This is a hydrophobic region of the enzyme. Turns out tyrosinase is a membrane protein, meaning that part of the enzyme spans a phospholipid bilayer. Where do you think this sequence that's full of nonpolar amino acids is located? And we should be getting pretty good at this by now. So here's our phospholipid bilayer, right? This is how we draw a phospholipid. We have a polar head and two fatty acid chains, which are nonpolar. So when you put the phospholipid bilayer together, this is kind of how they assemble themselves. You have the polar heads on the outside and the fatty acid nonpolar tails on the interior in the transmembrane domain. Here we have our extracellular domain and our intracellular domain. So we have to kind of think that we are zooming in, right? If I zoomed into this part of the cell, this is what I'm looking at. And then this is the outside of the cell. This is the inside of the cell. The interior of the phospholipid bilayer is here. Okay, if you have a nonpolar region, where would you put it? Would you put it with the nonpolar hydrophobic part or hydrophilic? This is water. You are a sack of water inside and outside. And most of us got that it was in the transmembrane domain, because it's in the nonpolar region, because the sequence is hydrophobic. Okay, many proteins span the phospholipid bilayer in alpha helix formation. Remember, we have primary, secondary, tertiary, and quaternary structures of protein. The alpha helix is a region of local structure based on hydrogen bonding. What level of protein structure is alpha helix? It is secondary. That's where we have our hydrogen bonding, and it's local folding. Here we have some math. What we're telling you is that each amino acid is 1.5 angstroms, a really, really, really small unit of length. So each one of these is 1.5. This is 1.5 angstroms. Then we tell you that the bilayer is about 30. So this part is about 30 angstroms thick. If you had a bunch of stuff that's 1.5 long, 1.5 angstroms, how many would it take? How many amino acids do you need to span the phospholipid bilayer's transmembrane domain? So what you would do is you would do 30 divided by 1.5, and that gives you 20 angstroms. Good job. If an amino acid change rendered the enzyme totally unable to bind tyrosine, do you think the phenotype, think about the appearance of the organism, would change? It just so happens that we are talking about a metabolic pathway, that ends in the production of eumelanin, which is a pigment in skin, hair, whatever. And here we're talking about, in the signature assignment, we're talking about Siamese cats. Okay, I don't really care if you mentioned cats or not, but if you now make tyrosinase completely useless, can you make eumelanin? No, you can't. And if you're not making melanin, then the appearance, the phenotype, would change. Yes. Okay, how? You may have a loss of pigmentation. Okay, in the Siamese cat, they have the dark colored ears and their nose and their extremities, basically. Oh my gosh, am I going to even attempt to draw a kitty? Yay, kitty! Okay, so they have like dark end of their paws and their ears, and that's because the expression, just so happens, the expression is temperature dependent. Okay, so yes, if you had an enzyme that no longer worked, can't bind to tyrosinase, can't bind tyrosine, then you can no longer do this pathway and end in eumelanin. Now we're showing you the whole biochemical pathway. Okay, we showed you a very simple biochemical pathway. We showed you a very simple enzymatic reaction to start with, maltose and alpha glucosidase. Now we're showing you a little more complex one that involves phenylalanine and tyrosine to start with, and then we use some various enzymes to end with either eumelanin on one side of the biochemical reaction or epinephrine and norepinephrine on the other side. And this is adrenaline and noradrenaline, basically. And we're asking you to predict what would happen to the levels of each substance if you were to remove the enzyme AADC, or aromatic L-amino acid decarboxylase, from a cell. So here's the decarboxylase right here. Let's say you got rid of that. Just a broad question. Could you make any of these? No, you couldn't. If you no longer had your aromatic decarboxylase, you could no longer synthesize dopamine, norepinephrine, and epinephrine. But you did not affect tyrosinase. You could still synthesize all of these. So we have to look at each substance. Did we change the amount of phenylalanine by affecting AADC? No, we still start with the same material. So phenylalanine, is right here. Phenylalanine stays the same. Okay, stays the same. Same with tyrosine. Tyrosine stays the same because we didn't do anything to this enzyme, phenylalanine hydroxylase. We're still producing tyrosine. Okay, so that stays the same. Norepinephrine and epinephrine decrease because we got rid of our aromatic decarboxylase. And then if we look for L-dopachrome, here it is, and eumelanin, let's think about this. If we had, this is the example that I was giving in class, if we had water flowing this way and we split it into two, now we have a blockage over here. What happens to this flow? It would increase, right? You're starting with the same amount. You decrease this amount here. Bam, you decrease that because you're no longer able to make dopamine. So now you have an increase on this side of the reaction, and you're going to increase eumelanin and L-dopachrome production. So those two, oh boy, increase. Bam. Okay, if you have questions on this, or if you didn't score a 20 out of 20, please feel free to correct it and re-upload it, and I will grade it again.